Hello everyone and welcome to our webinar today. My name is Beth Turner. I work for the NOAA National Centers for Coastal Ocean Science. I'll be moderating the webinar today. Jennifer Mintz from the NOAA Ocean Acidification Program is our producer. She's going to keep Adobe Connect going today. If you're having problems with Adobe Connect, you can enter problems into the chat box on the upper right corner of your screen. We'll try to address them. I do note that if you're having problems with the audio, you might try logging out and logging back in, especially using the application rather than the browser. We found that that gives you a better audio experience. Thanks for joining us for the effects of ocean acidification on HABs, a review of what we do and don't know. This is number 7 in a series of webinars to prepare for our virtual HAB OA workshop. The webinar series is laying the foundation for the virtual workshop, which will be held August 11th to 13th. And we're going to discuss HAB OA interactions, the major questions surrounding them, and research priorities that can help address the gaps in our understanding and management of these factors. The workshop will help us identify priorities and research products that can be incorporated into a future request for proposals. If you're not yet signed up for that workshop but you'd like to participate, please contact Maya Sharp. Jen will put Maya's email into the chat box. Maya, I'm going to let you introduce yourself and share a little bit about your summer project with us.
Thank you, Beth. Good day and welcome everybody. My name is Maya Sharp. I am a NOAA Educational Partnership Program undergraduate scholar, and I'll be completing a project based on webinar's content, questions, and discussions. The chat box will be monitored and recorded during the webinar. There will also be a questionnaire that we will use to inform our workshop and future directions of research funding. Those accessing the webinar will have the opportunity to comment, ask questions through the Google form. The link will be in the chat box for discussion at the workshop. I will compile these and synthesize them for our workshop. Thank you.
Thank you, Maya. Jen here, and I just want to take a few moments to get us all oriented in Adobe Connect. As you all may have experienced, audio is over the computer, which means you can listen through computer speakers or a headset. If you are having any trouble hearing, you may try either turning up the volume on your computer speaker or headset, or you can turn up the volume in Adobe Connect. That is something you can do in the top menu bar by clicking the pull-down arrow to the right of the speaker icon and selecting Adjust Speaker Volume. If you're on a headset, you may go to the speaker icon, hit that pull-down, and select Speaker to find the headset or speaker that you are looking for. As Beth mentioned, if you're still having trouble hearing, please go ahead and log off and log back on, using the Adobe Connect application rather than the browser. During our presentation today, you are welcome to view it in full screen, and you do that by heading to that button with the 4 arrows in the upper right edge of the slides. This button toggles slide size larger or smaller. You would want to note that you'll lose chat and captioning boxes in full screen, but can toggle back to that regular smaller view at any time to ask questions or add comments into the chat box. We do ask that you enter your questions into the chat box highlighted here. We'll hold all questions until the end of the presentation today. And as Beth mentioned, we'll be sharing relevant links, including the questionnaire that Maya mentioned, that we are asking all participants to take a few minutes to fill out after the presentation. At the end of the presentation, we will open it up for questions and discussion. If you would like to add a comment but are not the questioner, you are welcome to raise your hand using the icon highlighted here, and we will go ahead and unmute you, and you can have that conversation with our presenter today. We will post a recording and a PDF of the slides on the workshop webpage. The link can be found in the chat box, along with that link to the questionnaire, which will be found on that website for viewers to send follow-up questions and comments to Maya and the workshop planning team after the webinar. We can't emphasize enough that you take a few minutes to fill out that Google form or questionnaire so that we can use those responses to make for a robust discussion at the workshop and also inform directions for future research. Beth, I will hand it back to you.
Thank you, Jen. Today we are welcoming Melissa McCutcheon, who is a PhD candidate in the Coastal and Marine System Science program at Texas A&M University in Corpus Christi. She works in Dr. Jinping Hu's Carbon Cycle Lab. She's hoping to defend her dissertation later this year, and for that she is examining spatial and temporal variability in the carbonate system of Texas estuaries with a focus on the physical, biological, and geochemical controls on the estuarine carbonate chemistry. She completed a 6-month NSF-funded internship with Ocean Conservancy in December 2019. During her time there, she completed an extensive literature review on the impacts of ocean acidification on harmful algal blooms, and that's what she's going to be presenting today. So thank you for being with us, Melissa, and take it away.
Thank you. Can you hear me?
Yes.
Okay, great. So I'm really honored to be invited to speak amongst such a great lineup of scientists for this workshop. As she said, my background's mostly coastal carbonate chemistry, but I had the chance to delve into this interaction with HABs while I was interning at Ocean Conservancy last year. So I just wanted to recognize Sarah Cooley and Ryan Ono for they really came up with this question and guided me through this investigation. And so I wanted to also recognize NSF non-academic research internships for graduate students, supplemental funding opportunity, because they funded that internship and allowed me to delve a little into the policy world and into what I'll be sharing with you today. So just to outline what I'll be talking through with you today, first we'll just have a little introduction. Most of the time I'll be talking about the physiological effects of ocean acidification on phytoplankton and HABs. And then I'll also talk a little about OA and phytoplankton community composition, and then finally get to some conclusions and discussion. So I'm sure most of you have seen a figure like this. Atmospheric carbon dioxide concentrations have been increasing since the start of the Industrial Revolution, and with that, 30% of those anthropogenic CO2 emissions have been taken up by the ocean. So ocean surface waters are increasing in CO2 and subsequently decreasing in pH. And that has the potential to negatively affect a lot of different marine species. But acidification can actually be intensified in some of the coastal regions because of substantial spatial and temporal variability in the carbonate system driven by physiological, biogeochemical processes such as freshwater inputs, nutrients, organic matter loading, stratification, and upwelling can also come into play in those regions. And so harmful algal blooms are basically naturally occurring algal species that are allowed to proliferate under certain environmental conditions, causing these large blooms that can cause concerns for human health or the marine environment, including different organisms and fisheries, and can have many negative effects. And so they've kind of been on the incline for several decades. Looking at this paper from the early 90s, you can see that they were already saying at this point that HABs have increased in frequency, intensity, and geographic distribution for the last couple of decades. And so, we know that they're detrimental to humans or the environment. They also kind of vary by region, and so the types of HABs that tend to pop up are different among different areas, and each have, these different detrimental effects. So many produce toxins that may either be directly tied to human health concerns or may be toxic to fishes, birds, or marine mammals. But they don't necessarily have to produce toxins to be harmful. Sometimes just their dense growth can lead to problems like hypoxia and anoxia and subsequent marine die-off. And so there's been a lot of investigation into this growing concern of HABs and how it overlaps with the suite of different environmental stressors that are occurring at the same time. So increasing temperature, stratification, ocean acidification, eutrophication, all of these things may interact with HABs. And you can see that there's been some investigation into all of these. I really like this review figure on the right here from Wells et al. 2015, clearly illustrating that a lot of these environmental changes seem to be correlated with growth in certain HAB groups, but you can see much greater confidence for a lot of the other stressors outside of OA. So, there's still that uncertainty as to what ocean acidification does to this, and despite that OA is unlikely to be the driving, main influencing environmental factor, it definitely has the potential to interact with these other factors and to compound their effects or, worsen HABs in general. And so I'll be trying to go kind of from the general to the specific throughout the presentation. So I'll be talking about OA and other factors and how that affects phytoplankton as a whole, and then how they affect HABs specifically, and how that may be manifested in phytoplankton communities. So I like this figure, which is kind of highlighting that the potential for increased CO2 will allow natural kind of cyclical blooms of phytoplankton to become greater in intensity, and the environment can support greater phytoplankton populations under those conditions during bloom. But, again, relatively little is known specifically for HAB species, what will happen. And so briefly I wanted to talk about the motivation, for this literature review and what I did at Ocean Conservancy looking at this HABs and OA linkages. So, Ocean Conservancy basically focuses on providing the right information to policymakers, for them to be informed when they're voting on legislation or budgets or funding scientific research or environmental management. And so given the impacts of HABs on food security, tourism, the economy, and human health, legislators usually do care about this issue. And so, I like how Ocean Conservancy makes this information really digestible. And so this infographic here is showing that somewhere between $2 and $24 million is spent annually in healthcare costs that are associated specifically with red tide events in Florida. So that kind of highlights the importance of this. And also, basically based on linkages between these different environmental stressors, you can gain additional support for policy decisions. So this map I created also during my internship, this is looking at basically the concern for environmental issue of ocean acidification by each state, and that's reflected in how the states have taken action or invested for ocean acidification. So you can see here the colored states have introduced legislation mentioning OA, dark blue having passed legislation mentioning OA, and you can kind of see, you can kind of think of this as the state's perceived importance of the issue. And so while areas that are currently experiencing most exposure to acidification do seem to align with those states that are taking some action, there are still states that have a lot to lose if they are hit by OA that are not really acting. So for example, a lot of the Gulf states do not currently show much concern by this proxy for ocean acidification. However, several of them have in recent years been hit pretty hard by harmful algal blooms. So Florida and Mississippi specifically, have had these issues over the last couple of years. And so from a policy standpoint, it's going to be really important to highlight the linkages between these different coastal issues to bring legislatures on board with progressing that policy. And just as one example of the importance of kind of intertwining these different stressors in coastal environments, scientists are already looking at this complexity and how things overlap, and will continue to do so. But policy even is taking this holistic view, and so this piece of federal legislation that passed the House last year that concerns funding of ocean acidification research actually specifically mentions in text, incorporating multiple stressors and looking at how that interacts with harmful algal blooms. So with that intro, I'd like to go ahead and get into this discussion of the physiological effects. And so two different main physiological effects to phytoplankton that I want to discuss are how ocean acidification may change growth rate and how it may change toxin production. Now, both of these are going to be important to the overall effect of the harmful algal bloom in the environment. And something that I also wanted to point out is that OA itself can already kind of be considered multiple stressors. So, the concentration of carbon dioxide is changing in the water at the same time that pH is changing. And, many investigations of the effects on phytoplankton are really looking at mechanistically how the change in PCO2 is going to change their physiology. But fewer have looked at pH. And I did want to say, pH could be important because it would affect, maintenance of intracellular pH, membrane transport processes, phosphorus and trace metal acquisition, since those are tied to the bioavailability. And so, it may also be relatively important. But, getting into the discussion of growth rates, we see that, the main thing that's been discussed is the increased CO2 in the water and how that will affect growth rates. And so different phytoplankton become saturated with CO2 at different levels. And at what point they become saturated is usually going to depend on whether or not they have a carbon concentrating mechanism and how it functions. And so in this figure, you can see that what is labeled as pumped is what is the carbon concentrating mechanism. And it is basically responsible for increasing the affinity of CO2 for the photosynthesis process. Because photosynthesis is generally CO2 limited, since rubisco, the supporting enzyme, has low affinity for CO2. But this pump allows inorganic carbon to be delivered to the location of the photosynthesis, and it's usually accompanied by another enzyme that will convert inorganic sources of carbon into CO2 that can be utilized. And so, this plays a really big role, but I think just as important as its presence is its plasticity. So some organisms are able to regulate the way in which this functions, and if there is an increase in the total amount of carbon available, they can downregulate and redirect the resources, the energy that would be used to other physiological processes. Uh-oh. I think that I did something wrong and my screen went away.
Hold on 1 second, Melissa. We'll see.
I'm so sorry. Okay, great. I see it again. So sorry about that. So to continue, again, we're talking about growth rate and how that may be affected by ocean acidification. And so we see that species and even strain-specific responses have been recorded for this. And so I'll talk about that a lot more. Okay, so the next thing, again, we're going to be looking at how ocean acidification may influence toxin production. And so basically, there has been a report of increases in cellular toxin production in some HAB species in response to ocean acidification. This has been tied to element availability in the environment. So toxins seem to be produced in greater quantities in response to elevated CO2, tend to be the carbon-rich toxins, whereas toxins might actually be produced in greater amounts due to limited nutrient availability if that nutrient is not a large part of their structure. And again, we see species and even strain-specific responses here that we'll talk about a lot more. So I'm going to be working my way through a series of slides that are showing basically the results of the literature review that I did. So I basically tried to find every relevant study that investigated possible changes in growth rate or toxin production in response to acidification. So this table that you see in the top right will be coming up repeatedly to display the findings for several different taxonomic groups. So in the boxes beneath growth rate or toxin production, you'll see numbers that show up. Those are representing the number of studies that showed evidence for a physiological response to OA, either in the positive direction, which would be found in this box, in the negative direction, which would be found in this box, or it was tested but no response was seen. And I'll talk through some of the examples. I won't have time to go through all of them. I also want you to keep in mind while I'm going through this that many of these studies were laboratory monocultures. So of the 24 different studies of different HAB responses to OA that I found in the literature, only something like 5 were competition experiments instead of monocultures. So therefore, many did not actually represent the complexity that would be found in a community. Also, the majority of the studies do not actually capture the presence of evolution in a HAB species because they're more short-term. So those are things to keep in mind with this. And we can kind of think of the numbers, as a proxy for the confidence that a certain physiological response will take place. So first, I want to get into cyanobacteria. So this is blue-green algae. They're basically found in regions, as far as marine regions, they're found in coastal areas with substantial flooding because they're usually found in really low salinities. They actually contain very efficient CCM with the highest affinity for carbon dioxide, which makes them really successful in low CO2 concentrations. But also they're believing that their CCMs are very plastic, and so displaying that plasticity would mean that they would also potentially function well under high CO2 conditions. And so here you can see the results in the table. There's evidence of one study showing that microcystis will have increased growth rates in response to ocean acidification. 2 studies found evidence that microcystis will have increased toxin production in response to ocean acidification. So, we do kind of see some variance in the results here. For example, one competition experiment, a toxic microcystis species was able to dominate over non-harmful green algae, more so in high CO2 conditions. However, another monoculture experiment with microcystis found that non-toxic strains became dominant during CO2 conditions. So, it's still up for debate how they may respond in ocean acidification conditions. Next, I'll talk Correnia brevis. You're probably familiar with this. This is the dinoflagellate that's responsible for Florida red tide and neurotoxic shellfish poisoning. So, really prevalent throughout the Gulf of Mexico. And here we see there is evidence from one study showing that they will increase growth rate in response to acidification, evidence from another study that they may not, and no evidence yet for increase in toxin production from this group. And here's just one example of a study that showed when comparing low and high PCO2, Correnia brevis was able to switch their inorganic carbon preference from 14% to 56% CO2 used. This is basically showing that they do have that plasticity that allows them to maintain the same carbon fixation rate and growth rate across different PCO2 levels. Here is another example from the literature. This experiment basically is one of those that looked at toxin production and did not see that there was an increase in cellular toxin production. So per cell, we did not have more of the toxin present, but because this study found that the growth rate was elevated, the overall amount of toxin that was produced, which can be seen over here in your figure on the right, was increased by acidification conditions. Okay, the next group that I want to move into is Alexandrium. So if you're not familiar, this is the dinoflagellate that's responsible for paralytic shellfish poisoning. It's pretty widespread across the West Coast and does have some occurrences throughout other places in the continental United States. And it has been kind of the most studied. So there's been 5 different species studied. And what we see looking at those responses is very variable within this taxa. So, 4 different studies had evidence of increased growth rate in response to OA, but we also see evidence of decreased growth rate and so on. So I'll talk about that just a little bit more. Here is one of the several studies that looked at this organism. Basically, these experiments were conducted with different strains that were isolated from different locations, but it was the same species within this taxa. And you can see that the different experiments found differences between strains in the response to ocean acidification in both growth rate and in their cellular toxicity. So here is an example of those strain-specific differences. There was also another study that looked at 8 different Alexandrium strains within the same species and found that just 2 of them were stimulated by increased PCO2. So we see this kind of across the board, that this genus seems to have a lot of different responses between species and even between strains. And then just one more example for this species. This is kind of showing how ocean acidification may interact with other environmental factors. So in this study, they found that in an N-replete environment, the toxicity was unaffected by changing CO2, but in an N-limited environment, the toxicity was increasing with increasing CO2. And so, but we do see that the N-replete environment across the board tended to have higher cellular toxicity. And now here's, some more condensed part of the review where I'm looking at several different taxa in one slide. You can see these different organisms have had fewer studies examining them, whereas these diuretic shellfish poisoning species did not have any noted response to ocean acidification, but these other HABs, Carlidinium, Cochleodinium, did have increased growth rate and in one case toxin production. And we can see that example from the literature. They were actually found, in the same study, Prorocentrum and Carlidinium. You can see that we did not see that response for Prorocentrum. We did for Carlidinium, and that's also reflected in their cell ratios. So they were grown together. Okay, Pseudonychia is another really important harmful algal bloom that people are concerned about. This is a diatom responsible for amnesic shellfish poisoning, producing domoic acid, and it has pretty widespread occurrence along the west coast, also Gulf of Mexico and East Coast. So what we see for it is there are several studies that showed that in response to ocean acidification, there was an increase in growth rate or toxin production, but there are also conflicting studies. And here's just one example from the literature showing that toxin production increased with increasing PCO2, especially under phosphorus-limited conditions. Okay, one more taxa to delve into that has been investigated. This Heterosigma, we've seen 4 different studies that show evidence that there will be increased growth rate in this organism. This is an organism that likely does not have a CCM, so it makes sense that they would respond positively to increases in carbon dioxide concentration. And here you can see what that looks like from one of the studies. I just wanted to point out here that it took several days for that response to level off. So this kind of shows the importance of having kind of longer term studies. This seems to level off after 7 days, where you could see that that increased growth rate was sticking around after that point in time. So in grand summary, if I compile all of the different harmful algal blooms, we see that 16 different studies showed evidence of some type of increase in growth rate in response to OA. 10 showed evidence of increase in toxin production in response to OA. So there is a strong potential that some HABs will increase in both their frequency and toxicity. However, I wanted to point out that there are several different important HABs, taxa, that are not present in the literature. So for example, we didn't see anything about brown tide, didn't see anything about gymnodinium, which is another PSP-producing organism, Gambier discus, that's associated with ciguatera fish poisoning. Physteria has caused huge fish kills in the Chesapeake. None of them have been present yet in the literature, so that's something that's really still lacking. Okay, and I'll try to really quickly go through this section, looking at how communities might be important. So this is something that I think that Beth Stouffer's presentation did a really good job of looking at, but I kind of wanted to highlight this meta-analysis from 2015 that showed that changing carbon dioxide concentration is expected to impact the total phytoplankton community function more than any other anticipated ocean changes, such as warming, stratification, or nutrient availability. And so, you know, because of these differences between different functional groups, and because they're going to be interacting with each other in the environment, ecosystem dynamics, including competition, predation, and viral infection, may be just as important as the other physiological responses. And I liked this study that was basically looking at a total phytoplankton community and seeing what the difference would be in that community between control conditions and high CO2 conditions. And we know, a lot of HABs are dinoflagellates. So while this study was not specifically looking at HABs, if we look at the dinoflagellates, we see that they did not experience increased biomass in response to elevated PCO2. But what I did want to highlight is that they did bloom earlier and have an overall extended bloom period. So there's still potential that that changing community composition could have impacts on the effects of harmful algal blooms. And so the real question becomes, will harmful algal bloom species outcompete the other non-harmful algal bloom species? And so this meta-analysis is brought up by several of the presenters so far, and it basically suggests that most HABs have a positive growth response to ocean acidification, while most non-HABs do not have a growth response to ocean acidification. And so, by that logic, we would expect potentially that the HABs could become more successful and more competitive under more acidic conditions. This is another good study that kind of looked at that altered competitive fitness of HABs. And so it was looking at microcystis and found that microcystis, toxic strains of microcystis outcompeted 3 species of green algae under those high PCO2 conditions, while green algae was able to outcompete that toxic strain under lower PCO2 conditions. So again, acidification conditions may make harmful algal bloom species more competitive. And so the final thing, I just want to kind of go through conclusions of the review that I've done and talk about grand challenges for research by this community in the future. And so for conclusions, first, given that literature review, given all of the findings of all of the studies that have been conducted thus far, there is strong potential that some HABs will increase in frequency, intensity, and toxicity in response to ocean acidification. The exact physiological responses may vary between species and even between strains. And that's also going to be influenced by other environmental factors. So differences that have been detected thus far between species within the same larger taxonomic group, or even strains within the same species, they may be real inherent differences, or some of those differences may be due to other environmental conditions like nutrient or temperature variability that may not have been part of that experiment. So that is going to need a little bit more investigation. And then finally, ecosystem dynamics, such as competition, may prove to be important in determining the competitive success of harmful algal bloom species as ocean acidification progresses. And so just some grand challenges that I wanted to talk through. So these grand challenges, I think everybody so far during this presentation series has presented some really good ideas about what the real challenges to the community will be. These are going to be in context of the topics that I've covered today and in context of the literature review that I've done. So first, I do think that we do need to continue filling in the gaps at a species level, looking at those changes in physiology, because we still have taxa that are completely missing from investigation. We still could look more into the influence of multiple stressors. So, despite that other speakers have pointed out that physiology is really what we are best informed about as far as this OA-HAB interaction, I think there are certainly areas that still need attention within this. So, based on the review that I've done, I think that we still need to continue filling in those gaps. And even, those species that have none or only one study, it's possible that some strain-specific differences that we've seen already, they mostly occur in Alexandrium, which is also the most well-studied. So because we see the most variability within the species that is best studied, that might suggest that further investigations would be warranted. Next, we, of course, need to broaden our understanding of the effects of OA and HAB species as they occur in a full community assemblage. So we need to look at different trophic levels. We need to look at multiple types of phytoplankton in a community. And we need to kind of figure out how much of what we know about an individual in culture can help us piece together the total ecosystem story. And so it's going to be important to define how large and all-encompassing do these experiments really need to be to understand those phytoplankton communities. And then finally, this is kind of more coming from the perspective of Ocean Conservancy and thinking about why they would really want me to compile all of this information and their ultimate goal. It's going to be to support policy and management positions. And so, we need to be able to propose regionally specific water and land use management strategies based on evidence for coastal water quality outcomes, including HABs and OA. And so, first we need to be able to get to the point that we're able to propose these regionally specific management strategies. And so with that, I would like to open it up for any questions and discussion.
Thank you very much, Melissa. We do remind you that there is a Google survey that goes with this webinar. Jen is going to put the address for that Google survey in the chat box. And if you have questions, please enter them into the chat box. I see that Libby is putting in a question for her. Maya is going to read the questions as they come up. And Maya, why don't you start with our first question?
Thank you, Beth. Our first question is from Libby. She asks, has anyone done research trying to tease out the independent effects of pH versus CO2 on HABs?
Hi, Libby. Thanks. That's a good question. Honestly, from everything that I've read, I don't really see anybody trying to tease out those differences yet. There's a little bit that has been talked about oxidative stress, which may be more induced by changes in pH, but honestly, most of what's out there is kind of mechanistically explained by the changing carbon dioxide concentrations, and so they have not really tried to tease out those differences.
Thank you. Our next question is from Beth Turner. She asks, do many of the studies even report what the full carbonate parameters are, or just PCO2?
From what I've seen, Beth, it looks like mostly studies are just reporting PCO2. Alternatively, there are studies that just report pH and the response to pH, but don't necessarily talk about it as much physiologically or mechanistically as those studies that looked at PCO2. But yeah, I do not see a lot of studies, reporting saturation states or that full parameterization of the carbonate system for sure.
I believe I just saw another participant typing, but the question did not come up as of yet.
Yeah, it looks like Astrid might have been typing. Astrid, if you'd want to speak, you can raise your hand and Jen will unmute you if you want to say your question rather.
Yes, we have our question from Astrid. She asked, I was wondering what the general consensus was in regard to PCO2 as a driver versus temperature.
You know, it seemed to me that there were more studies that showed that temperature was a more important driving factor, at least in growth rate, than PCO2 was. However, when you're looking at the meta-analyses, the one meta-analysis suggested as far as community function, because different taxa are hit differently by PCO2, that it may be more important. So, I don't know that a full consensus is there yet. I think there have been more studies looking at temperature as a driver than there have been looking at PCO2 as a driver, for sure, but not even a ton of them that are looking at them in conjunction with each other at this point.
Thank you. And Astrid says thank you as well for your answer. And we have Jesse Turner typing the question, so we can wait for that. We have a question from Libby. She asked, I'm wondering whether we have models now which connect CO2 and changes in HAB abundance in United States waters.
I believe that is something that has been brought up during one of our previous webinars. It's not something really that I'd read a lot about during this literature review, honestly, but I think it is something that's underway. I don't know how robust those are right now.
Thank you. And our next question is from Jessie Turner. She said, newbie question, What are the regional specific actions for managing HABs and reducing impacts? I assume efforts related to controlling local contributions of pollution, nutrients, and etc.
I think that you know, your assumption is probably correct. You know, locally, acidification can be driven by nutrient input sources, not necessarily by increasing atmospheric carbon dioxide concentrations. And so if we know that certain areas, certain eutrophic locations are kind of responding to those nutrient loads, the regionally specific management plans may be to reduce those nutrient loads into that location. And you know, that is going to help with multiple different stressors that are occurring, both the acidification and the harmful algal blooms in many instances. So, you know, that's kind of something that we need more information to directly know, like, what level of nutrient load reduction is required to see improvements.
Thank you, Melissa. And if anybody would like to comment, we can unmute them just by raising your hand.
Okay, it looks like the questions may be tapering off. Please raise your hand or type in a question if you have one. In the meantime, we're going to post a recording of this webinar on the workshop website, which Jen put into the chat box. I remind you again to provide your responses and any comments on the Google form that's available on that website, and encourage you to fill that out, as we do use that for our future funding decisions. Our next webinar is going to be this Wednesday, July 15th at 2 o'clock Eastern Time. Chris Holderried from the NOAA Kasutsina Bay Laboratory will present on Alaska ocean acidification and HABs, networking, and coastal variability. Thanks for joining us today, and we hope to see you on Wednesday.